Cells make up all living organisms, and in this lesson we're going to introduce cellular structure and then also function of those different parts of the cell. Now, as we start to talk about cells and what they're actually going to be doing as far as function goes, there are 4 very important processes that are required for all living things. And this is what really is used to classify something as being living. First off, they need to be able to grow. Growth and then also reproduction. They would need to be able to give rise to new individuals like themselves. Another one is that they need to be able to respond to their environment in some way. So they need to be able to respond to changes in their environment so that they can survive ultimately in the end. And then lastly is that they need to be able to metabolize a variety of substances. When we talk about metabolism, it is essentially run chemical reactions. So they need to be able to grow, they need to be able to reproduce, they need to be able to respond, and then again, run some chemical reactions. The organisms come in lots of different sizes. So this ranges from the large animals that we have, large plants that we have, all the way down to the microbes. And when we're talking about microbes, if you just compare them to, in this picture, to a chicken egg, a microbe, it could be a protozoan, which are going to be your larger ones. And then we also have bacteria, which are significantly smaller. And then viruses are going to be even smaller than that. Viruses, though, I will mention here, are not considered to be living by most individuals because a virus is not able to do all of the things that we see in this list. So again, for something to be considered living, they need to be able to conduct these 4 main processes. When we do talk about cells, which is our focus here in this lesson, there's 2 main types of cells. There are prokaryotic cells, and then we also have what we call eukaryotic cells. Prokaryotic cells are going to include most of our microbes. They are organisms of 2 different domains of life, archaea and then also bacteria. They are really, really tiny organisms. When we get to the eukaryotic side of things, eukaryotic, these are organisms that are found in the domain Protista, fungi, Animalia, and then also Plantae. And you can see in the pictures here, these are going to be much, much larger organisms. Typically, they're going to be multicellular organisms, so they're going to be made of many cells, which is one of the reasons why they are ultimately going to be much larger organisms. Now, size of the cells. Well, a prokaryotic cell is much smaller than a eukaryotic cell is going to be. And it's also going to have a lot less components when we're talking about the prokaryotic cells compared to the eukaryotic cells. We're first going to focus on the prokaryotic cells. Prokaryotic cells, again, include bacteria and then also archaea. So let's talk a little bit about the structures of bacterial cells, and then we'll talk about the archaea cells. Around the outside of a bacterial cell is where we find the external structures. These external structures is really anything outside of the membrane. So we will have a membrane, and then there's these extra things. First up on the outside of the bacterial cell is going to be the glycocalyx, or if we're talking about it in plural, it's glycocalyces. So we're going to start on the outside and move our way in. So this would be the very outside of the bacterial cell. It is a gelatinous, very sticky substance that is going to surround the cell. This does fall into 2 categories. We have some that are called a capsule, and we have some that are called slime layers. And if you look here, the capsule is going to be a thicker substance. The slime layer is going to not be quite so thick. It's not going to provide as much of a layer on the outside of the cell. So what is the function of having this sticky substance on the outside of the cells? What is the function of the glycocalyces? They have a few different functions that we can list. One of them is going to be, first off, that it does protect from desiccation. Desiccation means drying out. So by having this extra layer on the outside, the cells are not going to be as susceptible to drying out or dehydrating. Another function is that this is going to provide a means for attachment. This will help that cell to attach to a variety of surfaces. It also is going to help multiple bacterial cells attach together if they do need to attach together to form a colony or a biofilm or some other structure. And then lastly, this does play a role in helping bacterial cells in some cases to avoid recognition by a host. So this will help them to evade the defense systems of host organisms that they might be infecting. Another external structure that we have with bacterial cells is the flagella. Flagella are kind of like long tails. This is going to be a long structure, a long filament that extends beyond the surface of the cell, and it will actually penetrate and go out of the glycocalyx. It has three main parts to it. It has a filament, which is the long tail component. It has a hook, which is going to help with the rotation because we're going to be spinning this around. And then it also has what we call a basal body. The filament itself is composed of a protein called flagellin. That flagellin is where the term flagella comes from. It's just because it is made up of flagellin. And again, this filament and flagella is supposed to spin like this. Now, if we look at types of flagella, there's lots of different types of flagella as far as lengths go and how many you might have on the surface of a bacteria. We have what are called peritrichous flagella. Those are kind of all over the place, and so we can see that in the first picture here. That particular bacterial cell has flagella covering the surface. Sometimes they have just one. That's called a single polar flagellum, and then sometimes they have a few that are coming out again from the same location. So on the bottom there, you can see where they have a few different tufts, as we call them, polar flagella. And those are coming out not only from a single location, but it's from one of the poles or ends of the bacterial cell. There are some unusual bacteria that are called spirochetes, and these have spiral flagella. And these spiral flagella happen to be located at both ends, and the fact that they're at both ends provides kind of a corkscrew type movement. So the movement of the cells is a little bit different from what we have with the typical flagella that we find in bacteria. And one thought is that this corkscrew movement, it might help them to evade some defenses of the organism a little bit. So the spirochetes might, because of just the way that they're moving, have a better defense mechanism. Now, in general, if we're talking about how the flagella works, what it's going to do is that it's going to be powered by ions kind of gushing and coming out of the membrane. So this ion kind of squirting or gushing out of the membrane will cause turning of this entire flagella. The flagella is going to rotate 360 degrees, similar to the way that a propeller would rotate. So just think about if you blew air on a fan or on a propeller, that would cause this rotation like that. That's what the gushing out of the ions is going to do. And these propellers, these flagella, they move really fast. So they can move up to 100,000 RPMs. That's revolutions or turns per minute. So 100,000 turns per minute. This is going to enable them to move pretty fast. And when they move with a series of what they call runs and tumbles. And the runs are when they're moving pretty fast, so it's like a constant movement, and all of the flagella, if you have a bunch of them covering the surface, they're kind of rotating in the same direction. And then the tumbles will be like a sudden change in movement direction, sudden change in the direction of rotation, and this causes them to move kind of randomly and stop moving so directionally. So they'll go directionally for a moment, that would be their run, then they switch the direction that their propellers are rotating, and of course that causes them to kind of jolt to a stop, and that is the tumble. So what is the function of the flagella? Well, one of the functions is movement. It is what we call taxis, which is movement in response to some type of a stimulus. So they would be wanting to move towards that stimulus or maybe away from that stimulus. And we describe the taxis in different ways depending on what the stimulus happens to be. If it is movement in response to light, whether it's to the light or away from the light, we call it a phototaxis. If it is movement in response to a chemical, we call it chemotaxis. And then as far as towards or away, that's where we have positive taxis or negative taxis. So positive is when it's a good stimulus for them and they're moving towards that particular stimulus. So we can have a positive phototaxis, we move towards the light, or we can have a positive chemotaxis and we move towards the chemical. Or we can have a negative taxis, which is when they want to move away because it's not something that they want to be near. It's very unfavorable. So if we have a negative phototaxis, we're moving away from the light. If we have a negative chemotaxis, we're moving away from that particular chemical.